This tutorial focuses on submission of environmental sequencing data and metadata to the European Nucleotide Archive. The European Nucleotide Archive, the ENA, is a permanent and comprehensive archive for public domain nucleotide sequences and associated information. The ENA provides the following services to the user community: archiving, help desk, training, standards for development, technology development, and also community building. The ENA, GenBank in USA and DDBJ in Japan exchange data daily as a part of the International Nucleotide Sequence Database collaboration. We will now very briefly explain how is environmental sequencing represented at the ENA. This will allow better understanding of the ENA data model, which is helpful for users submitting data as well as users retrieving data from the European Nucleotide Archive. Environmental sequencing typically comprises several steps. At first, a scope of the environmental study is formulated. Subsequently, samples are extracted from the environment and then sequenced using shotgun sequencing or amplicon-based sequencing. Afterwards, reads and/or sequences are generated. Finally, read assemblies, sequence annotation, and/or data analysis are produced. Description of the sequencing project is captured in the study object. Sample information and contextual information on surrounding environment of the sample are archived in the sample object. Information on how the DNA or RNA sequencing experiment was performed is held in the experiment object. Machine-generated raw read data are described in the run object. Assembly information, annotation of sequences, or data analysis are archived in the analysis object. In summary, the ENA data model consists of data, which are raw reads and nucleotide sequences, and of metadata, which is information associated with the molecular data. Metadata describe provenance of a biological sample, a sequencing experiment, its scope, its analysis, and raw data reads. Every object of the data model needs to be described in a standardized way in order to ensure the whole sequencing project is meaningful and reproducible. This section will explain how to register samples associated with the sequencing study. Sample registration means description of the sample and of the environment where the sample has been extracted from. In order to facilitate sample description, the ENA has developed a number of checklists, which are basically pre-templated forms with control vocabularies and support of relevant community-agreed reporting standards such as MIXES, microbial or barcode. The following demonstration will explain step-by-step registration of a sample associated with molecular data. A molecular sample can be registered or submitted using the Webin submission tool. You need a submission account to use the Webin submission service. Please create a new submission account. For any help, please contact datasubs@ebi.ac.uk. This created account can be used for all subsequent submissions of all users associated with the account. When you receive your submission account details, log in into the Webin. Once you enter the Webin system, select New Submission tab. Now select the option button Register Sample. Navigate to the ENA checklists using the Next button. You are now on a page listing all available ENA sample checklists. ENA checklist simplifies sample registration into 3 steps. Step 1. Select suitable checklist. Step 2. Select appropriate descriptors of the chosen checklist. And Step 3. Add values to the selected descriptors. On this page, you can accomplish the first step. Select the appropriate checklist from the menu, for instance, the ENA microbial checklist. Note that this page will also allow you to upload and complete the checklist from a previous session using the Upload Spreadsheet button. If you don't have a completed checklist, go to the next page. On this page, you can accomplish the second step, to select appropriate descriptors of the chosen checklist. The right-hand side of this page lists all mandatory fields of the checklist. Additional optional fields can be added by selecting appropriate optional descriptors from attribute groups on the left-hand side of the page. The optional attributes that you have selected will be added to the right-hand side of this page. For instance, you may wish to provide concentration of nutrients in the water body. Then please click on the plus button of the attribute group Mix as concentration measurements and select nutrients you measured and you wish to provide values for. Complete information on the right-hand side of this page. Note that this page contains information which is common to all your samples. In the section Organism Details, start to type in the organism name. If the organism name is classified at the NCBI taxonomy, it will appear in the list and can be auto-completed. For example, for marine environmental samples, type in the search field Marine Metagenome. This will automatically add the correct NCBI taxonomy identifier to all your sample records. Select relevant sampling campaign from the pull-down menu. Provide sampling site. Type in the site name. If the sampling site name is included in the site registry of the OSD sampling sites, it will appear in the menu and can be auto-completed. Provide sampling platform. Complete now all remaining information common to all your samples. For this particular checklist, this means to add common values for fields in the attribute groups Event, Sample, and Environment. Go to the next page and using the Add button, specify the total number of your molecular samples that you wish to register. Add the sample group details. By providing information to fields on this page, you will complete the third step, which is to add values to the selected descriptors. Note that this page contains sample-specific information. Data common to all samples added in the previous step are already included. You have now two options to add sample-specific information. First option is to use Webin interface directly to add data to each sample. We will now use this option for demonstration purposes and complete sample-specific information for the first sample. Second option is to download the checklist in the form of a spreadsheet to your local disk using the Save Spreadsheet button. This spreadsheet will contain all mandatory and all your preselected optional fields. At your convenience, complete all information locally and then start a new session, that means log in again into Webin, and upload your spreadsheet using the Upload Spreadsheet button. When information for all samples is completed and valid, you can submit your samples with the Submit button. Submitted samples will receive the ENA accession numbers that will be linked to reads and sequence data.